We start? Yeah. Good afternoon, everyone. Thank you for joining for another webinar edition, which ABLE is conducting with the support of BASF. As all you know, as most of you know, like Association of Biotechnology Enterprises is one of the premier pan-India organization. We have been conducting various programs in support of biotech, as well as we have been coming out with lot many reports, so which we update regularly. In the process, as you must have seen, we have conducted many webinars. This is one of the important webinar which we are conducting today with the support of BSF. We in this webinar we have two speakers joining from BSF, and we also have Dr. Murali, past president as well as current president of Council of Presidents. So Dr. Murali will provide you will set up the context as well as tell about the ingredients, that is how we plays very important role in production of biologics, as well as we have speakers who will be talking about ingredients from the industry point of view, how it is going to make a lot of difference in terms of either yield or production, all those things. I request Dr. Murali to please take over and set the context for us. Thank you.
Thank you, Bala. Thank you very much. I hope I'm quite audible to everybody here. So welcome to the two lovely ladies here today. So we have Dr. Aditi Poddar, Business and Technical Service Manager, BASF Pharma Solutions. And this is part of South Asia. That's what I understand. We have Dr. Nadya Morales, Global Technical Marketing Manager, BASF Pharma Solutions. So for people who may not know about BASF, I would say then you're in the wrong seminar hall. But if all of you know about BASF, then you're in the right place here. They are one of the most illustrious companies in the world. And we have two ladies talking about a very, very important topic. India is now poised. It's almost doing a lot of work on the biopharma. And all these things have a lot of challenges, as you know. I mean, every single organization strives for quality, and there is no compromise on that because there's zero tolerance to anything that's not comparable, compromisable, or anything like that. It's not possible at all. And the more illustrious the companies are, the more careful on how they would like to see that their supply chain, their ingredients, their analytical methods, and all those things that are very, very important for maintaining a uniform and a consistent quality across the globe is so important. While this is a very regulated segment, it still needs such a care. And a lot of the companies and a lot of people who are part of this seminar know how valuable it is to ensure that there's a huge supply chain which goes from one end to the other end. It's not so easy to switch. We need to be very sure that all of them are complying so that, all the regulatory part is taken care of. And so we can't have a very poor product quality. We can't have, low performance. We can't have any of those. So there's absolutely zero tolerance to any one of this. So these two ladies are now going to take us through on what is that BASF is doing and what is that they are going to tell us that we can learn today on a Friday afternoon. Normally in Europe, this is beer time, but I guess this is, I'm talking about a Friday afternoon with some cheese and balls and all that. But anyway, it's okay. For the rest of the people, I think it's going to be after the seminar. I'm guessing the order is where Nadia will come first and then Aditi will come next. If I have said the order wrong, you can do it in the way you both ladies want to do it. The floor is all yours, and we are all ears listening to what you're going to say. Thank you very much.
Thank you. Thank you, Dr. Murli, for the introduction and setting the tone. So, I will be taking you through the first part of the presentation, but before that, I would request Dr. Nadia to please introduce herself a little more. We'll get to know more about her. So, over to you, Dr. Nadia.
Good afternoon to all of you. As my colleague has said, my name is Nadya Morales. I am the Global Technical Marketing Manager for Biopharma Ingredients, part of BASF Pharma Solutions. I am today I am in my home office. It's quite early in the US, but my lab is right outside New York City. And in my lab, what we do is test and develop data that I'm going to be sharing with you a little bit later on how our ingredients can impact bioprocessing. I am very excited. This is my very first time giving a webinar to Indian audience. So I am very touched for the invitation and very excited for the opportunity of showing you our data and sharing with you some of what our ingredients can do for you.
Great. Thank you. And also, I'll introduce a little bit more about myself before I start presenting my presentation. So, yeah, this is Dr. Aditi, and I have around 11 years of experience, and I have a doctorate, and also I am mainly solely from the pharma background. Here in BSF, I am basically taking care of the technical services of the South Asia region, and yes, as part of this duty or responsibility, I take care of new customer projects, troubleshooting of certain formulations, and generating new customer relations. So, yes, looking forward to good interactions. And yes, I will now share my screen so that we are now good to go for the presentation. Please give me a moment.
So just to save some bandwidth, some of us will switch off our videos so that you can go ahead with it.
Aditi, you're in mute.
Is the screen visible?
Yeah, you can. It's visible. You make it a slideshow if possible.
Yes, definitely. I'll just switch to the. Is that okay now?
We can't see the slideshow.
Yeah.
Now I have put it in the slideshow now. I believe it will come soon.
Okay. Yeah, it's not on the full slideshow yet. Yeah.
Not yet.
No, we don't see the presentation screen. We see the individual
Okay.
slide.
Okay, just let me reshare my screen.
Are you using a single screen, Aditi, or are you on two screens? Because if you're having two
No,
screens,
I'm
sometimes you
on
have a
single
problem.
screen.
Okay.
Now, how is it looking?
Now
Is it in the...
can,
I'll just shift to the presentation mode.
yeah.
Is that fine now?
Now it's still on the other mode, but I think we are able to see it. So if you can sort of go ahead, there's nothing wrong with that.
Okay, because I can see it in the presentation mode. So
there's another way you can do this. If you can send it to Indrila, she can also project it from
Yes,
her side.
I have it with me. If there is no change in the
yes,
slides, I can share it.
yes,
Yeah,
that will be great.
Indrila, please do it. That saves a lot
Yeah.
of problems.
Yes,
Thank you. Thank
Yeah,
that
that's
would
you.
be
perfect.
great.
Thank you, Andrea.
So Aditi, one of the things is that whatever technology we may have even a supercomputer in front of us. But remember, when we do a webinar, there is something that will definitely go wrong. So you don't have to worry about it. This is how it is.
No. This is how it is.
Yeah, great. Go ahead and you can
Good,
say
good,
next
good.
slide, please. Next slide, please, so that she
Yes,
can.
yes, definitely.
Is it on slide mode?
Yes, absolutely.
Okay, fine.
Thank you.
Thanks.
So yes, thank you so much. Well, good afternoon once again to all the audience who are present here, and thank you, Abel, for giving us the opportunity to present on BSF as well as the product that we have for biopharma in particular. So I will take you through the presentation that is in general talking about what BSF is, who we are, and what different business units we have, how do we work, and what are the different principles and specialities that we have in the pharma area. And then in the later part, Dr. Nadia will help you understand the more in depth about the biologics and the ingredients that we have that is required for different processes in biologics. So if you can go to the next slide, please. Thank you. So, if we have to talk about BSF, we are a big multinational company. As you can see, that we have around 1 lakh or over 1 lakh employees around the globe, and we do see that we have a very good sales in the 2024, that is 65.3 billion. We have around 235 production sites, and we do have a concept of Verbund. Now, what is this Verbund? Verbund is basically an interconnected setup of manufacturing units, wherein the you can consider a manufacturing unit A, B, and C, for example, and the byproduct of manufacturing unit A becomes the starting material for manufacturing unit B. So these Verbunds are interconnected, and hence what we see here is a very unique concept wherein the material or the usage of material is quite optimized, and the idea is quite sustainable, so very less wastage of any kind of material that is being generated in a manufacturing unit. So yes, we do have these 6 urban sites, and as I mentioned, we do have a very good sales and 235 production sites. If you can go, please go to the next slide. So these are basically our operating divisions. So we have materials, agricultural solution, nutrition and care, industrial solutions, surface technologies, and chemicals. In general, if you talk to any person who is, not knowing BSF much from pharma perspective, they will always correspond that BSF has a lot of agricultural solutions. So BSF is known more for the agricultural solutions in general that I have seen. We are known for a lot of the insecticides, the fungicides, the enhanced seeds, etc. So yes, that is one of the biggest business unit or operating division that we have. Other than that, yes, we have nutrition and care, industrial solutions, surface technologies, and chemicals. We as pharma, we fall under this category of nutrition and care. So next slide, please. So, in case of nutrition and ingredients, we have certain sub business units, which is also called as the global business unit. One of them is the human nutrition. Human nutrition business unit develops or basically is related to the all the vitamins that we have, vitamins, carotenoids, and omega 3. So, this is a very big platform for all the vitamins that we manufacture. Then we also have the animal nutrition. In case of animal nutrition, we have certain offerings like other vitamins, then carotenoids, and also feed enzymes, which go into the animal nutrition part for the, I would say, the domestic animals like cattle, cows, poultry, etc. Next slide, please. Then we have the aroma ingredients. This is, as the name suggests, it is all related to the fragrances. So we have unique offerings, basically citrus scents, lavender scents, mint scents, and also around 40 products that are, kind of made with a mix of these fragrances. And yes, this is also a very big business for us. And the next slide, please. And then finally, yes, we have the pharma solutions. So in pharma solutions, we have a lot of offerings or products that can go into different usages, and also certain active pharma ingredients and certain virtual pharma assistant facilities also. So maybe we can see that more in detail as we proceed through the presentation. So next slide, please. Yes. So Pharma Solutions, who we are, what we do, we'll see more. So next slide. So if we see, we will be discussing more on who we are, what are our solutions, what do we do with respect to digitalization, and what we are in, what is our stand with respect to sustainability. We also are, as I mentioned, part of this BSF group, and we are part of this new human nutrition and health under this BSF group. So can we go to the next slide? So if you see who we are, in general, for excipients, we have always thought ahead of time. So when we think of tomorrow's excipients, and we do have certain facilities or certain offerings in a digitalized service also, and we do have a very long track record of bringing in new innovative excipients, APIs into the market. So yes, for us, innovation is one of the most primary thing that always comes to our mind. So yes, we are in this field for a very, very long time. Along with that, we have certain digital solutions. So I will discuss that more in the upcoming slides. So next slide, please. When we talk about the services, that can go into three important categories. One is the quality, then the regulatory, and the technical services. So if you see the quality per se, as Dr. Murli also mentioned in his introductory talk, that yes, for us, quality is very important. Quality is something which is non-negotiable, and hence we take a lot of pride in saying that our products are of very high quality. And we also offer a lot of third party audits. We also offer certain certifications to our products, and that enhances, or that keeps a more big basis for our quality. Then regulatory, we do have, as I mentioned, a digital platform. In that, we have one platform called as Reg Excellence, which is more like a 24/7 available platform for all the users, from wherein they can download all the type of documents that are required for a pharma understanding. For example, it can be a manufacturing process, a specifications, or any kind of certifications that they need, etc. Then the technical services. So we do have a good network of experts all around the globe, wherein I also being part of this network. So I represent the technical service from South Asia. And yes, we have all the expertise required for providing customer services, and also not only the expertise in person in case of personnel, but also analytical and good sophistication that we have. So we try our best to give the utmost good service to the customers via a lot of technical troubleshooting, case studies, application data, etc. Next slide, please. Yes, again, for us, innovation is top priority. Whenever we think of something that we can do for the customers, or something that is new to be launched, yes, we are always ahead of time. And as I mentioned, the concept of urban, which is quite unique to BSF. And yes, we do a lot of manufacturing which is under the GMP, because this is, of course, it is going into the pharma space. So yes, GMP is something which is very important for us, along with superior quality and the concept of sustainability and good reliable supply. Next slide, please. So if you see the presence of BASF Pharma in general, you can see the star that is mentioned, that is the headquarters. We are based out of the US. And then the red circles that you see, the red font, that is our major production sites. Major production sites being for ibuprofen, PVP, PEG, and also the lipid-based excipients, omega-3. These are all our major offerings. I will also take you through some of them, I mean, I would try to cover these excipients in the upcoming slides. And the blue ones are basically our R&D and tech service and the business development locations. So if you see the blue ones, we also have one tech service lab in Mumbai, and the others are based out of Shanghai, Germany, US, and South America. The one that is there in Germany, that also is the R&D center, and that's where the major innovation and the thoughts that are there that happens in the R&D lab. We are there to support the customers. If you also have any queries with respect to development or generation any application data or any other queries that you have with respect to formulation, we will be more than happy to support you through these application lab that are there. So yes, Mumbai would be the best choice, take given the presence in India. Next slide, please. So yes, as I mentioned, our solutions that can be categorized into different subcategories. So we have offerings that are there that goes into oral solid dosage form, certain that are helpful for solubilization, topicals means skin delivery, and of course the biopharma ingredients, which we'll be talking more in the next session. Also, we have the APIs and the parenterals. Next slide, please. So for orals, we see that we have a lot of offerings that can go into different applications. For example, it can go into tablets, mini tablets, capsules. We can also have offerings that we can go for ODTs, granules, pellets, powders, syrups, suspensions. So more or less, we have offerings that can cover majority of the solid oral applications, and also the products can help us modulate the release profile. For example, if we want instant release, or we want taste masking, or we want to have enteric release, sustained release. So all the product offerings that we have can cater to these release profiles also. And not only that, as I mentioned, we have the thought for futuristic concepts. So our products are also suitable for certain next level technologies, like we can say 3D printing and also continuous manufacturing. So all in all, if you see in orals, we have a very, very broad product portfolio that can cater to almost all the solid orals and all, as well as the liquids that are available. So for orals, we have, I will reiterate again, we have lot of excipients for tablets, we have disintegrants, binders, granules, pellet for the instant release, and also the coating materials, matrix formers, etc. So next slide, please. Yes, for solubilization, this is again a technology that we can support. So as of now, we all know that many of the APIs that are being synthesized or coming into the market, they are poorly soluble in nature. And to aid the solubilization, we do have certain products that can help in solubilization. So the solubilization can be done by majorly two techniques. One is formation of solid dispersions. So for solid dispersions, we do have offerings that can go into techniques like hot melt extrusion or spray dried technology. And do we also have product offerings that can go into lipid-based drug delivery system? For example, we have a lot of surfactants, co-surfactants, and co-solvents that can help in the formation of this lipid-based drug delivery. So yes, again, for solubilization, we have a lot of offerings in our product portfolio that can help for both solubilization in the solid as well as liquid oral formulations. Next slide, please. Then the topicals, we do have a lot of offerings for the topicals, that is, be it creams, be it gels, be it lotions. And if you see, we have a lot of offerings that can go as a viscosity modifier, it can go as a structuring agent, it can go as an emulsifier. So yes, we have options that can go into the traditional dosage form, as I mentioned, like creams, lotions, gels, and ointments. And it can also go into emerging technologies like transdermal patches, foams, or films. Again, when we think of our products, they can help in delivering the active ingredient to the required site of action on the skin, be it a local action or a systemic action. We do have certain, we lot of ingredients that can help in this developing this formulae. And also we have certain studies done on these products that help us to understand whether they have a mildness, because as we know that for skin applications the product has to be mild. It should not be allergic in its own being, and it should be not create any side effects on the skin. So these mildness studies also we have done at our end to understand and see to it that our products are quite mild in nature. And also the sensorial experience that we see for most of the products, be it spreadability, the texture, how the cream has a shine, these all are quite superior for the products that we have. So we do have a lot of as I mentioned, emulsifiers, structuring agents, gelling agents that can go into the solution. Next is, can we go to the next slide? Thank you. So we have the biopharma ingredients. So in this, Nadia will also touch base a lot on detail on this. So we have the upstream, downstream related products. So if you see, we have a shear protectant on which we will be discussing more on the upcoming presentation. Then we also have products that can go into the downstream, that is the purification part, and also a lot of stabilizers for the formulation. So be it stabilization surfactants or any other ingredients. So we have that for the formulation. So yes, for biopharma, we have solution for all the three major processes, be it upstream, downstream, or the formulation. Next slide, please. Yes. So we also have a lot of APIs in our product portfolio, be it ibuprofen, omega-3, L-menthol. So these we have been having for large period of time, and we do cater to a lot of customers for L-menthol and ibuprofen. Omega-3 also for pharma as well as Nutra applications, we have omega-3 available. Next slide, please. Yes. Now, again, as I mentioned, for certain parental applications, we also have the excipients that are required for parental formulations. That is for solubilization, formation of solutions for polysoluble API. We have a lot of options for parentals also. Next slide, please. Okay, so coming to other topic, this was all about the offerings that we have. Coming to the other topic, that is the digital solutions. I'll go. Can you please go to the next slide? So in digital solutions, we have three important uh, services that I can say. One is Zoom Lab, the other is Reg Excellence, and the other is the My Product World. In case of Zoom Lab, it's a AI-based tool where you can use it for simulating or developing initial set of formulations before going to the lab. So this is a huge database which you can use to design your formulations based on your need. The Zoom Lab will give you suggestions for your formulations. In Reg Excellence, as I mentioned before, also it's a repository of all the documents, and it can be used at the person who has registered can use it at their own disposal whenever they want. They can download the documents. And My Product World is another important site or a website that we have under Virtual Pharma Assistant, wherein we can find our products, and it is generally used by supply chain colleagues to understand where the what type of product listing we have, where is this tracking of the supply, etc. Next slide, please. So yes, this is how we offer the different digitalization. We using our My BSF World. You can use it to download the documents, to place an order, and we will be there to help you out. Next slide, please. Yes, so this is another important part of BSF's concept or BSF's thought process is the sustainability. For us, the target for 2030 is 25% reduction in carbon emission from scope 1 and scope 2. What is scope 1 and scope 2? We will see it in the upcoming slides later on. And 15% carbon dioxide emission reduction for scope 3.1. And by 2050, the aim is to go net zero in all the scopes for carbon dioxide emissions. Next slide, please. So we all know that through pharmaceutical industry, there is a lot of carbon emission that is done, and this is what we think it's a small way to contribute by reducing our footprints, by reducing our carbon dioxide emission. This is the only way where we can think that we can make an impact on sustainability and hence on the environment. If you can go to the next slide, please. So this is what I was mentioning about what is scope 1, scope 2 and scope 3. So scope 1 is basically the direct emission that we see from the industry per se. So for example, if BSF is manufacturing, scope 1 would calculate or understand the emissions from BSF's manufacturing site. Then scope 2 will be more of from the supplier or what kind of electricity that is being used for the manufacturing, and scope 3 will be all the subsidiary who are helping in this manufacturing. So for example, the raw materials that are coming from other sources for the manufacturing. So this is what scope 3 would mean. So in case of pharma in general, for API there's a lot of contribution to the carbon dioxide emission through scope 3. So we have to be very mindful, and BSF is quite mindful of how to reduce this contribution from scope 3. So we also are mindful of how we purchase the raw materials, from where we purchase the raw materials. Next slide, please. So this is how another important concept is the product carbon footprint. So considering scope 3, scope 1 and 2, this calculation is made, and this calculation comes to a figure which helps the customer to decide what is the sustainability based thought, or what is the sustainability that is there, whether this product is sustainable or not. Using this product carbon footprint, we can understand. So customer, as a if they are very conscious about sustainability, they will always look out for this product carbon footprint whenever we are whenever they're dealing with any vendor. So yes, this is an important parameter, and we try to see that we have the least carbon footprint for our products. Next slide, please. A very good example that I can think of is about the production of ibuprofen. As I mentioned, it is happening in the Texas in Bishop. So we try to see it is as sustainable as possible. For example, if you see, we have the production steps that are reduced to 4 versus the number of production steps that is seen for other supplies. It is 5 to 7. Also, it is produced with 100% green electricity, so there is a 10% PCF reduction altogether. So all these steps have helped us to synthesize or manufacture ibuprofen in such a way that it is totally sustainable and there is a reduced environmental footprint. Next slide, please. Another big example is for one of our product called Colidon 30. So BSF is the pioneer in this PVP chemistry. We are the one who have discovered PVP and also the poloxamer chemistry, and Colidon 30 is one of our biggest or highest selling product for BSF. Now, if you see the manufacturing, how it is manufactured, again, it has been manufactured via Verbund concept, and hence, as I mentioned before also, that this Verbund concept helps in aiming towards sustainability. So yes, this is sustainable, and because of that, the carbon emission that you see, or the footprint that you see from the manufacturing of BSF by BSF method, is less than 30% than the industry average. In fact, it is much lower than what we see for other competitors also. So this is a big achievement for us, and we believe that this is something that we should be, quite proud of, and hence our Colidon 30 is quite sustainable. So I think this is all about it. So this is what BSF can offer for you with respect to sustainability. The PCF certification is available for certain products, and the thought of sustainability is always there in our mind whenever we think of manufacturing a product. So yes, I think if you go to the next slide, yes, over to you, Nadia, and thank you for your patient listening.
Thank you so very much, Aditi, for that introduction. So I am going to be in charge of the more technical topics, as Aditi mentioned before. Today, Aditi mentioned that we have ingredients for the different stages of bioprocessing. However, today we're going to be focusing on that upstream part, and we're going to be talking about our product, our Poloxamer 188, which is used as a shear protectant in the bioproduction industry. So here's an outline of the topics that we're going to be talking about. I'm going to start with what is the impact of variability of raw materials in the bioprocess, and do a short introduction on what Poloxamer chemistry is. So if we can go to the next slide. So raw material variability, especially for those of you who have been either in the biomanufacturing or even the greater pharma industry, you know that variability in raw materials is something that is very undesirable. Here on the slide I have on the right side, I have a graph. It is from a reference that actually talks about, it takes all of the ingredients that are involved in making in the media that is actually made for growing cells in biomanufacturing, and they actually put them in different buckets. And you can see here they have the hydrolysates and the surfactants, which are both very key ingredients of these kinds of media, are the two biggest problems when it comes to variability of the raw material. And why is it very undesirable? So of course, it can change the cell performance, right? But it can also, and more importantly, impact the quality of the final product, right? And why is there variability in these kinds of materials? Some of them, I mean, even though the industry has tried very hard to move away from animal source materials, even chemically synthesized materials can be, because of the different chemistries, can still be very variable and have changes in composition. So we are going to be talking about one of those surfactants, which is Poloxamer 188. If we can go to the next slide, please. So Poloxamers, it's actually a chemistry that BASF invented back in the early 1960s. And it is actually a very versatile chemistry. I always describe it as a sandwich. So essentially we have the slices of bread are the parts that are ethylene oxide, and they are hydrophilic. And then the filling of the sandwich is propylene oxide, which is the hydrophobic part of the Poloxamer. And in this chemistry, by changing the amount of those two ingredients, by changing those proportions, then you can change the kind of properties that the material has. So if you actually have been part of the pharma community and are a little bit perhaps know a little bit about BASF, we actually have been offering these kinds of chemistries for many of our other pharma applications in tablets, in topicals. And you actually may recognize some of them here, the colisols, the different coliforms. But these kind of chemistries, even though they're great and versatile, they were never really meant to be hard in use in biology, right? In biological applications. So these actually offerings that we have on the right are the new offerings that BASF has that are the same chemistry, but we actually had to make a little bit of change to them so that they can be fit for the purpose of biomanufacturing. If we can go to the next slide, please. So what is the problem with, I mentioned that the Poloxamers were never meant to be used in biological applications. They were very much invented to try to help, especially in solubilization of poorly soluble drugs. However, for the biomanufacturing industry, there was found that especially P188, Poloxamer 188, was found to be a great solution to shear protection. So I mentioned initially that there has been a shift in the market to go into chemically defined medias. If you remember, like me, I was a student a very long time ago. Many of our medias when we were in school had animal ingredients, and one of those animal ingredients was serum. In serum, so we used to have horse serum and bovine serums. And one of the jobs of serum, well, of course, it gives growth factors, but it also was a shear protectant. So when there was a shift to change into chemically defined medias, there was a lot of looking for ingredients that could actually fulfill that shear protection job. And poloxamer 188 was identified as a surfactant that could help with that. But what was the problem? As I mentioned, poloxamers were never meant to be biological, right? They were never meant to be in this kind of application. And of course, we as in BASF, we are the biggest manufacturer and supplier for these kinds of ingredients, and we were making them for other applications that surfactants are usually used, not only for pharma applications, but also for home care. So many of our customers in biomanufacturing started buying that type of material, and they encounter a lot of variability anytime that they got a lot. So sometimes they will put an order, they will get material, the material will work, they will run their bioprocess, they will have great yields. They will buy the material again, and then they would actually find that that lot was not great. So if you actually look at the literature between 2015 to about 2019, a lot of the papers and the titles of the references are very much look like the part on the right. It's a lot of effort in trying to understand, and our customers trying to understand what makes a good lot of P188 and what makes a bad lot. And of course, we don't want our customers to go through that. So if we can go to the next slide, please. So what is it that our customers were seeing? And perhaps if you work in biomanufacturing, the graph in the left seems a little bit familiar to you. So as I mentioned, this is from a reference in the literature, and where there was an investigation as to why this bioprocess that this customer was running was so variable. And here on the A frame, we are looking at percentage of viability. So the percentage viability of the cells. In each one of those little figures, like the little squares, the little stars, each one of them refer to a different run of the process using different lots of P188. And as you can see, if there was no variability in the process, each one of those figures would be right on top of each other, right? And that is actually what every bioprocess engineer strives, right? That every time that they run their process is repeatable, and it's the same, right, with the same yields. But as you can see, there was a lot of variability in the viability, and also there was a lot of variability in the amount of viable cells. So each time they run the process, sometimes a lot of P188 would yield really high numbers of cells, which means more product, and other times it would have a lower number of cells. And of course, that variability is unacceptable, and nobody wants it. So we actually partner with a customer, one of our customers in pharma, and where we have been their supplier for all of their solid dosages. And they say, hey, we're starting to offer products in biologics. And this material, the quality of this material is unacceptable. Can we work together so that you can actually bring a material that we can use reliably? And we said, sure. So what we did with our poloxamer is we changed some of the manufacturing controls so that we could actually minimize that variability that our customers were seeing lot to lot. We also eliminated some of the contaminants that actually gave this kind of variability. So as Aditi mentioned, we have manufacturing plants all over the world. Many of our manufacturing plants are multi-product manufacturing plants. So sometimes, especially for the poloxamers, as they have the same starting materials, many of the poloxamers are manufactured one after the other. And that's a problem, because inevitably there's going to be a little bit of that residue going into the next product that is made in the line. And for some applications, that's not a problem, but for biologics, that was a big problem, right? Cells are very sensitive. So we actually changed our manufacturing to remove those kinds of contaminants. And as part of our release, we had added analytical testing and application testing. So we have an HPLC method that we use. I'm going to show you a little bit. And we also have a cell test. So we test our materials using CHO cells before we release the lot to make sure that our customers are getting the most pure, fit for function ingredient. Can we go to the next slide, please? So now I'm going to tell you some of the optimizations on the product. So as I mentioned, part of the what is it that we changed? So how is it that it changed? So here on the top frame, we have a cartoon of a chromatogram. So we have a reverse phage HPLC method that we developed to to recognize if one of our lots had this kind of contaminants that I was talking about before. So many of these contaminants are others, maybe other surfactants that are very hydrophobic, and we know that cells don't like hydrophobicity a lot. So in this circle area part of the chromatogram, if there were any kind of contaminants, the peaks in the chromatogram will go in that circle. So we actually have made the changes that we made in manufacturing completely removed the hydrophobic contaminants that we had seen in previous products, in our previous product. And on the bottom, I show you also an example of how our cell tests look. So we actually, what we do in our lab, and actually the cell test happens in our lab. What we do is actually we try to simulate using a flask. We try to simulate very high shear conditions, and where we put our poloxamer, and then we measure both cell viability and viable cell density. And here is just an example on how that would look like. So initially, our product, the product that our customers were buying before we made our Coli4 P188 Bio, was that second one, the negative control, which is this light gray control that we have in the graph. That is actually what our old product used to give us for how the cells grew in that, using that product. And then the other four green bars, each one of them actually represent some of the lots, some of our commercial bio lots. I am very proud to say that since we actually made the manufacturing changes to this product and launched the P188 Bio product into the marketplace, we have not had one lot with any problems of any contaminants. So we are very proud. We're very proud to say we actually, it's almost going to be 10 years of this product in the marketplace, and we're very proud that we actually have been able to make this product every single time just as pure as the time before. If we can go to the next slide, please. I can show you how the cells look like growing in our product, and how they look like sometimes when our product has this kind of contaminants. So here on the left side, this is an SEM picture that we took in our lab. And on the left, we actually have CHO S cells that are growing. We took a sample from a culture that is growing in our P188 Bio. And you can see that we have great number of viable cells. The viability of the cells, it's very high. And our cells look round. You know, the cell morphology is exactly how you expect a CHO cell to look like. On the right, we have a product. So it's also a poloxamer product, but we actually spiked it with some of the hydrophobic contaminants that our customers used to find in our product. And you can see that what happens to the cell. The cell, it's completely shriveled. You can see that pretty much we don't have any live cells in this frame. And in many cases, some cells, of course, are more sensitive than others. So obviously, this is a very extreme case, but traditionally, we can see if there is any kind of hydrophobic contaminant in the poloxamer, we can start seeing decreasing cell numbers starting at about 400 ppm. If we can go to the next slide, please. So I spoke to you about the changes that we made with our first product, our P188 Bio, which was a big change in manufacturing. Once we started looking at this product in this kind of application, we realized very quickly that the product is not homogeneous. And of course it isn't, right? It's a polymer, right? So of course there is always a little bit of size distribution, right? A molecular weight distribution. And we started actually looking at what other parameters within the poloxamer could actually have an impact in productivity, in cell productivity. And one of the things that we found, and actually this customer found that we were looking at the same topic around the same time. This is why I always use this reference. This is a reference from Eli Lilly. And they noticed the same thing we noticed. So this is in this reference, Eli Lilly is transferring one process from one of their facilities to another facility. And so they're doing their trial runs, and they noticed that some runs, they actually had the normal antibody output that they were expecting. But other runs, they had a lower titer. And they were asking, well, why, right? So many people in the industry, they know that poloxamer 188 can be a problem. So they started looking at their different poloxamers that they had in their inventory, right? The different lots and different manufacturers. And they found that when they looked at the average molecular weight of the polymer, they were different, as you can see here in the bottom, in the left side in that table. There is actually a difference in the molecular weight of the poloxamer. And they saw that the poloxamer that had a lower molecular weight had better titers, as you can see on the right side. So on the right side is a chart, that one that is highlighted in blue shows that when they actually tried it, even at different concentrations, they have a much better, actually much better outcomes. And that is, they did this in these SP2 cells, they are B cells. We actually saw the same pattern in CHOs and in HEC293s. And let me show you those numbers. So if we can go to the next slide, please. So here is, this is our second product that we have in this line, which is the P188 cell culture. And what we did with this particular product is, so as I mentioned, it has a lower average molecular weight. And what we saw is that a really high shear condition. So we are trying, in the way that we test these cells, we try to replicate as much as it can be replicated in shake flask. We're trying to replicate perfusion speeds, right? So in perfusion, we have these very, very high shear forces. And what we see is that when we have those kinds of conditions, even though our P188 bio it's a great shear protectant, the lower molecular weight product, our P188 cell culture, outgrows the cells that we see when we grow them in P188 bio. So when you look at the viability, at the percentage of viable cells, it is actually, there's no difference. But when you look at the actual number of viable cells, the cell density, we have a significant difference. And we see this both in CHO cells, and we see it also in HEC293 cells. So if you are also starting to look at applications in gene therapy, it also has an impact on those and both cell grades. This is very product dependent. We actually have sent samples to customers, and some customers see great increase in cells for some of their product lines, and for others not so much. So this is actually something that it is very product dependent. So if you want to have a sample and try it in your system and see if it actually works for you, we would be very happy to sample it. What we also found is that not only, so it has an impact on the way that cells grow, but it also has an impact on the way that some of the products and the outcome of products. So if we can go to the next slide, please. Can we go to the next slide? Oh, thank you. So I'm going to give you also another example using gene therapy as an example. So in our lab, this year we had a big focus on developing our capabilities on being able to grow AAV adeno-associated viruses, which are used in gene therapies in our lab. So we established those capabilities this year, and we started testing our different grades of poloxamer. So we have our P188 bio, our P188 cell culture, and we of course benchmarked it against a third-party product. And if you're a little bit familiar, if you're not very familiar with the process on making these kinds of therapies, essentially we have 3 different plasmids that need to be transfected inside of the HEC293 cells to actually make the outcome of the virus. In our case, our virus make GFP, right? It doesn't make any real therapeutic. And we actually do, once we reach a height and where the cells are ready for bursting, we lyse all the cells to harvest the viruses, and we do an ELISA so that we can count the number of viruses. So if we can go to the next slide. So here's the outcome of those experiments, and again the comparison of the different grades that we have of poloxamer. So as I mentioned, in some very high shear conditions, our cell culture, our P188 cell culture product seems to have a great impact on the growth of the cells. For this particular application, it also has an impact in the overall production of the virus. So if you can see here on the left, we have the overall production, so the total titer of viruses. Our P188 cell culture is the one in blue. Our P188 bio is the one in green. And then we have a third party product that we benchmarked it against, which is in yellow. So not only do we have a higher overall number of viruses compared to our bio product and the third party, but we also, each one of those cells, the reason that we have so many more viruses is because each one of those cells is more productive when it's grown in P188 cell culture. Why? I'm working on that this year. So I'm hoping that maybe next year I can tell you more about why is this the case. But we found that when we looked at the overall differences in yield for this application, we see an increase of 64% when we use our cell culture grade when compared to the third party product, which is actually pretty remarkable, and something that when we were doing these experiments, we did not expect at all. So I'm hoping that next time that I speak to you, I can tell you a little bit more about why that is the case, because we are very curious to find out ourselves. The next topic, if we can go to the next slide, please, is about, so I show you that there is some changes in the cell. We actually wanted to start looking and understanding what those changes are. So if we can go to the next slide. So we did a bit of omics work in the cell, cells that are treated and cells that are grown in the absence of P188. In this case, we actually had, in this particular case, we have the comparison of our P188 bio against cells that were grown in the absence of poloxamer entirely. And what we find is that cells that are in contact with poloxamer. So poloxamer as a surfactant, we always thought that the way that it worked is that, and it does, it does incorporate itself a little bit, not covalently, into the cell membrane, making it more sturdy. But it also turns out that by being in the presence of the poloxamer, there are some transcription changes that we see in the cell. And most of the genes that are impacted are genes that are involved in the synthesis of the different subclasses of lipids that are recruited not only for the cell membrane, but also for the organelles that are within the cell. If we can go to the next slide. We also did this experiments, and where we were comparing our cell culture grade with our biograde, and we also saw a similar thing, in where when cells were in the presence of our cell culture grade, there was an increase that was significant on the total lipid content of the cell, and also it actually had an impact in having genes that are connected in the synthesis of sphingolipids that were also, that their transcription was also increased. So, and we are continuing, actually this year, we are continuing to do more work on this kind of content, because we want to try to completely understand what the impact on the whole cell is of these materials. But the reason that I wanted to share with you is perhaps you are part of the team that works on creating production cell lines, right? So most people use CHOs, right? But not two CHOs are the same. And I think that from the perspective of the molecular biologist, perhaps we don't always consider that some of the ingredients that we're using are they're not inert, that they actually can have an impact within the cell, and that perhaps that impact should be considered when we're making cell lines that are going to be used for production. So if we can go to the next slide. Other things that we have done in our lab is try to also understand I show you a little bit of the impact on the metabolism. Now I wanted to look more at the mechanical impact of the poloxamer in the cell. And what we found is that when we use anisotropy, which is a way of measuring, we use a fluorescent dye to measure the fluidity of the membrane. And essentially, we have this fluorophore that when the fluorescent light shines, if the membrane is really fluid, the fluorophore denses a lot and it actually emits a lot of energy. When it's more rigid, it actually has a lower, it doesn't dance as much, right? It's a lower resolution. And what we found is that in the presence of poloxamer that is fit for function, so and the green and the blue bars here are our bioproduct and our cell culture product, the membranes are more rigid. But here on the ones that are on the gray, we have the lighter gray is zero poloxamer, and the darker gray is poloxamer that has impurities. And you can see that the rigidity is lower. So we think that there is a combination between the recruitment that this, the higher transcription of genes that are involved in the synthesis of lipids, and also the mechanical impact of the poloxamer itself on the membrane itself, that in combination actually makes the cell more robust to be so we can actually grow it in culture. So I think that is my last slide. I think so. Yes, that is my last slide. I will be very happy to answer any and all questions. Thank you very much for your time.
Thank you, both Dr. Aditi as well as Dr. Nadya. It was very interesting. I'm sure people who are attending the webinar will be definitely would have benefited out of this. Now I request people who are there, if you have any questions, please post in the chat box. So our question box, we'll be able to take a few questions. And of course, I think they already shared the contact details. You can reach out to them also and probably take the conversation continue. So if you have any questions, we'll have plan for that. If any of the participants have questions, please type in your chat box. Meanwhile, just Dr. Nadya, based on your experience dealing with other countries, what potential you see in India for biologics as well as the kind of ingredients that is being consumed as an industry? So what do you think will be the future kind of thing? What you foresee or what potential we can think of?
Yeah. So I think that one of the things that actually makes the Indian market so exciting is because you have so many CDMOs, right, in India, and there is, of course, as we know, many of the very famous and profitable biologics are coming off patent. So there is that big opportunity for the biosimilars market. So I think that is actually where India is going to shine. And I think that this is a perfect opportunity to introduce ourselves and try to not only to showcase our products, but also to showcase that we have been within BASF for a long time. We didn't understand the impact of this product in this market, and now we're taking the opportunity to increase that knowledge and hopefully help our customers reach their higher potential. So that is I see my job very much as a educator of a sheer protection educator, so that customers can actually have the best outcomes. Because at the end, what we want is for patients to have access to those therapies.
Very good. Yeah, thank you, thank you. And if you can see, we have one question. One of the attendees is asking, what if the molecule are hydrophilic and need to make it hydrophobic and shear resistant after downstream processing? Is this possible with any of your ingredients? So what are ingredients you are processing? Whether it is, this question can be, you can take it up, probably Aditi or Nadya. You can see the questions also in your question box.
You know what? That is something actually we haven't thought about. I don't know. So our material is amphiphilic, right? So we have as I mentioned, the middle of the sandwich is hydrophobic, and the bread is hydrophilic. However, I will say that by changing the molecular weight, so I spoke to you about our cell culture product, which is a lower molecular weight. It is lower molecular weight. It's also a little bit more hydrophilic. So the opposite is also true. If you have a higher molecular weight, poloxamer 188, that is a little bit more hydrophobic, and that can help with that. I didn't show it today. I do have some data on that side, on the bigger molecular weight poloxamers, and we do see that they're great for, while the lower molecular weight is really great for growing the cells, the higher molecular weight is really great for formulating and for that downstream part of the process. So maybe that could actually help you with that piece as well.
Yeah, thank you. If anybody has questions, please post, and definitely we like to take it. Meanwhile, Dr. Aditi, you being in Mumbai, and we have a lot of startup activity, and a lot of startups are working on this area, developing biologics and biosimilars. And many of them have challenges in terms of getting ingredients, especially quality ingredients and all those things. So how do you think you can support them from Mumbai office, and how probably Nadya can also pitch in terms of if somebody wants to use the facility in other countries, for example, so whether it is possible for you to coordinate or you can help them to connect with those people.
Yes, definitely. So, as you rightly mentioned, that we have a lot of startups and a lot of small industries concepts that are, doing well in institutes as well as at a smaller scale. And if they need any kind of support with respect to these ingredients, we are ready to help them with the samples. We do have the samples available from our Mumbai office that I mentioned. We have an office in Navi Mumbai, that's in part Turbe, Navi Mumbai, to be more precise. So we can always support the customers with these samples for initial evaluation, and we are already doing it for a lot of customers, be it for pharma or biologics also. So yes, anybody feel free to get in touch with me, and we can always discuss on this.
Yeah. So we have any questions? I'm still due. Any questions? You can let us know. Meanwhile, Dr. Nadya, as you know that, we have been talking about a lot of biologics going off patent, and a lot of companies in India also scaling up or increasing the production capacity. So do you see any particular, trend that one has to catch up based on your interaction? Do you think that any particular trend in terms of either ingredients that is making a difference, or products that is going to see a good potential based on your interaction with other countries, as well as what you see probably from India point of view? And you have any particular interest or particular segment which you want to foresee a growth and you want to concentrate on that. Can you just elaborate on that if possible?
So, of course, I mean, we mentioned that the biosimilars, this is going to be a really big explosion in that market. But also, we also spoke about chemically defined medias, right? And that, I mean, the media is integral for the bioprocess. And of course, many of these medias, depending on the product that you're going to be making, it can have all the way to 100 ingredients. So another part of the industry that is really growing because it's needed. We need growth factors, right? We need to make many of the cytokines. So that is actually another part. I think that is the part of the bioprocessing market that is not spoken as much. That it also, so in order to make all of those ingredients, we also need to grow cells to make them. So that is actually, I think a really interesting part. And I actually had the opportunity of speaking with some of the startups, some startups in India, which are also focusing on making these kinds of, not making the final product, but making the ingredients that are going to help make those final products as well, which are just as important. So I think that is another area. India is starting to pick up on that. We see that very growing steadily also in places like South Korea as well, and where many of the companies, instead of focusing on making the biosimilar, they focus on making part of the ingredients, right? The ingredients that need to go into that media as well. So I think that is actually a part that is a little bit overlooked, but I think that many new companies are actually starting to focus on making high quality interleukins and growth factors that are going into that media, which is very valuable, as you would imagine.
Very good. Thank you very much for that inputs, because it's always nice to know what's from the outside, from people like you, what is the trend going on. We'll get a broader picture also. And we have another query where they're asking for the contact details of the speaker. I request both Dr. Aditi as well as Nadya to probably you can type in the chat box your contact email IDs so that they can reach out to you whenever they have anything to look after. Also, Dr. Aditi, while you type your email ID, I just want to know whether what kind of interaction you have with the academic institute, because many of the R&D is also happening at academic level. So what kind of support you can provide to academic institutions, be it in terms of collaboration or in terms of funding support? Anything is there? Please see whether you can elaborate on it.
Yes, definitely. So, as I mentioned earlier, also the lot of good ideas are, kind of being developed in the academia, especially in the IITs, and we have also ICT, which is Institute of Chemical Technology, and we also have certain other institutes for pharma, that is NIPERS. So, we always are open to collaboration with these institutes. So if they have any process or thoughts in mind, we will be happy to collaborate with them. And it could be vice versa also. Like if we have any ideas in our mind and it, kind of gels or sync with their thought process or their projects, we can do very well, develop nice projects or application data with these institutes. So yes, it's open. And we always have this thought in mind because whenever we think of the academia, their thoughts, their vision is way more, progressive, and they think a lot more, way more ahead than the industry per se. So yes, always open to collaborate with the IITs, ICTs there, and any other institute that is there in India.
Very good. Thank you very much. Of course, it is very important for collaboration is the one which is going to take it forward. Thank you for your, I think we'll have few academic participants also who like to explore collaborating with you. I mean, we are sure in coming days we will get few mails from them also. I mean, probably after seeing some of the recordings which we are going to share in social media and other places. Dr. Nadya, you have any special, what you say, message for some of your potential customers, like how you would like to approach them in terms of what you say, the product development stage? At what stage you like to join them, and how you want to I mean, not to say tread the journey in that way. So you like to just few words about what will be approach of approaching a protection customer? Like, India is, very cost sensitive. You know about it, so we don't have to tell you. So that being the case, you have any special plans for Indian customers where you can collaborate and then offer special services and other things?
Yes. We understand that the process of bringing any kind of therapy to the market is very long, complicated, and there's a lot of regulations. And we understand that perhaps when our customer or a potential customer started their process of making their product, they may have been using a different supplier. And we understand that changing suppliers can be hard. We understand all of that. We're very open to the startup that is looking for guidance on different ingredients. I have this process. Do you have something that can help me with that process? Either growing the cells or formulating it to get it to the patient. We can help. If you saw my presentation today, you see, maybe we can actually have. I mean, I used to use this other poloxamer from this other supplier, but I mean, the data that I saw today, maybe this is intriguing. We are happy to work with you too. As I mentioned, I see myself as the lead of the application lab. I see myself very much an educator. At the end of the day, of course, we are a supplier, and we would love for you to use our products. But in the end, we want therapies to go to patients. So if you think that we can help you understand your process better or understand the ingredients better that you're using, we are very happy. If you are local, please reach out to Aditi. She knows where to find me, and we can work. At the end of the day, I think that when it comes to these chemistries, the poloxamer chemistries that are used in this process, we really for a long time thought that it was just another ingredient in our list. And we continue to find that one ingredient that is so important can actually have other repercussions, not only on the outcome, but also on the cell itself. And I just want to make sure that our customers take that into account when they're engineering their processes, so that everybody is successful. So at any point in your design, either in the beginning or maybe you already have it, we will be very happy to talk to you, bring you samples, go over the data, and help you better your process.
Yeah. I'm asking a fundamental question because many of them will be participants. I'm sure will also have that question in their mind. I know you have. You must have heard from other people also. So basically, BASF is known for its chemistry and chemical-based products and services related aspect. And you now offering biologics-related ingredients and other things. Can you just can you give a story like how it all happened, like how you started working on this and what made you to come to this? Just to give a kind of history to that, so that people understand it's not something which has come as something like that, and probably some must have had some proper plans. Because in India, BASF is more known for, as I told you, for chemistry related aspect. Of course, Aditi, you have experience in it. So if we can throw light on that, probably participants will appreciate much more.
So I can tell you that BASF, so as you know, and as Aditi actually excellently presented, we are very much a chemical company. So we got into biologics it was actually because of our customers. So for a long time, when the biologics landscape started really taking off at the beginning of the 2000s, right? When their Humira, right? The Humira that is the big blockbuster of the industry started really gaining track and being supplied worldwide, was when we actually when our customers realized that there was a problem with this ingredient that they have been buying. For a long time, BASF was unaware until our customers came to us and said, "Hey, we're using this ingredient and it's just not working reliably. What is wrong with it?" And we were like, "But what do you mean?" So actually having that feedback from the customer made us realize that we were supplying this ingredient that we have been supplying for small molecule pharma for a long time, that it had a problem for this particular application. And so our customers coming to us and showing us the trust of saying, hey, this is not working really well. Can we collaborate to make it better? At the time when we launched our P188 bio product, my lab didn't exist. We actually collaborated with our customers. So we took care of the chemistry side, and our customer did all of the testing, the biological testing, to tell us, yes, this is the grade we need, this is the purity we need. So that actually then grew into our leadership understanding, hey, we cannot continue to be ignorant of this part.
Yeah.
Let's actually bring in experts. And that is actually how I came to my position came to be, my lab came to be, my team came to be. And since we started doing that, that is actually how we realized that we actually have underlooked this kind of chemistry in this application, that we actually have we never really understood exactly what it did and how it did it. So now we're hoping to catch on, right? So now that we are creating data to try to elucidate those answers, to try to continue to help our customers. And we actually have been very successful in that part, because now our customers, I think, trust us a little bit more. Like, they trust to come to us with problems. Another thing I wanted to point out to the audience is, we also, if you think that your process needs a different surfactant, BASF has we call it a cookie jar. We have the biggest cookie jar of chemistry.
Oh,
So if you think that there is a need for a surfactant, an anti-foam, something that you think that it could help your process go smoother, have better downstreaming. I know downstreaming is very expensive and very complicated. Reach out to us. We can collaborate, and we can look into different chemistries. I mean, as Aditi mentioned, our R&D is really focused on bringing innovation forward, and we have a lot of ideas. But at the end of the day, we're not the ones running the bioprocess. So we love to hear from our customers what your pain points are, so that we can actually help. So we can actually create products that are that can help not only them particularly, but also hopefully the industry at large.
Okay. Thank you. And I know we have a few minutes, just probably since we have no question, one last question to Dr. Aditi. I mean, apart from biologics, we also have cell and gene therapy segment also growing rapidly, and we have many startups as well as big companies investing a lot of time and efforts and money in the cell and gene therapy area also. So apart from biologics, whether your ingredients is applicable to those areas, or if you have any plans for that, or it is suitable for them also, you can throw light on that also, please.
So, as Nadya mentioned in one of her slide, our poloxamer, that is P188 Bio CC, both can be used for this cell and gene therapy, and it is being explored in that area, especially for the AAV and the HEK cell line. So we are into that. So yes, are the poloxamers that she mentioned, Colifor P188 Bio, P188 CC, both is suitable for be the biosimilars, be it via CHO cell line for MAP production, or for the CGT, that is the cell and gene therapy, via the HEK cell line. It is useful, and it has been found to be useful. Feedback has been received from customers, and also we have worked. I mean, Nadia has worked. Her team has worked extensively on the cell and gene therapy exploration also. So yes, it can be used.
Thank you very much. That's really it's like broadening your segment also. It's good actually.
Yes.
And we have one question, Aditi. I think you can take this in the question box. We say whether we can approach for cosmetic ingredients, wherein you take care of the chemistry and help us with the process development. So we have, as you know, Indian bioeconomy or Indian biotechnology includes nutraceuticals and supplements and cosmeceuticals, all those things. And since it is your company's chemistry based, probably that's where they're asking this question. Probably see if you can answer that.
Yes. So we do have, as I mentioned in my the initial set of slides, we have offerings for topicals, and that in goes for the cosmetic skin delivery. Also, we have a separate business unit altogether who caters to this personal care division, be it the cosmeceuticals or the surfactants that are required for making the cosmetics. So yes, if you have any queries, you can directly get in touch with me. If it's for a pharma application, of course, most welcome. We will be there to help you. If it's for a cosmetic application, I can direct you to our different business unit who is catering to this requirement for cosmetics especially.
Okay, so we'll wait. We have no more questions. Any last message you want to drop in for the benefit of the participants from both of you, both Aditi and Poddar, last word from your side?
I mean, from my side, first of all, I want to thank the audience for their time, because I know I'm in between you and your weekend. So I thank you so very much for your time and attention. And as I mentioned before, we are here to try to help get patients their medicine, however that may be. So if you think that our ingredients, our knowledge could be valuable to you, to your process, and to get better outcomes, please reach out. We are very happy to help.
Yeah, thank you. Aditi, you have something, you want to send a message to participants so that they can approach you faster, better, earlier.
Yes. So, first of all, thank you so much for your active participation. Thank you so much for posting the questions. And yes, we are there to support you for the requirements that are there, be it in the biologics. So, as Dr. Nadia mentioned, P188 Bio, P188cc is what we are offering for the upstream processes of biologics. Other than that, yes, as I mentioned in my presentation, also we have a lot of pharma offerings, solid orals, topicals, parenterals, APIs. So yes, you have my email ID in the chat box. So any query, please feel free to reach out to me. I will facilitate and see how best I can support you for all your queries. Thank you so much for your time.
Yeah, thank you, Dr. Aditi Poddar as well as Dr. Nadia for joining us for this webinar. It is, really interesting. We have good positive response from few of our participants, and I'm sure they'll carry a lot of learning from this and try to reach out to you as and when they have the requirement, especially for the ingredients what you have explained. I thank both of you as well as the BAF leadership team for approaching us and helping asking us to do this webinar. We are really fortunate to have that. At the same time, we're also, I also might extend my thanks to my colleague, Ms. Indrila. Of course, yeah, she was there back and managing everything. And also thank our president and other members who have been very cooperative and supportive of these sessions. And we are hoping to continue the dialogue with the BASF in terms of coming up with case studies and the other experimental results that is coming up, especially related to ingredients what you're talking about. We look forward to hosting you again very soon. Please, for participants who have participated, thank you very much for taking your time and attending the session. I'm sure you will have some feedback or requirement. Please do feel free to send the messages. We would like to see how best we can support you in that manner. Thank you very much for all of you to join. Good evening and have a nice weekend. Thank you very much.
Thank you. Thank you, everyone. Thank you. Bye bye. Have a nice weekend ahead.
Have a great weekend, everybody. Bye bye.
Bye.